So I'm just going to go now and start. In the interest of time and to make sure that we have a productive morning session or afternoon session, we are going to start with some talks this morning, the second day of CATMO, and we have some really good speakers this morning. So I'm honored to be the chair of the session. And the first person this morning is Diana Fusco, who is a physicist who had done her studies at the University of Milan before moving to the United States to do a PhD in computational biology at Duke, followed by a postdoctoral stay at the University of California, Berkeley, 4 years there, working on microbial communities, experience which allowed her to secure a faculty position at the University of Cambridge in the UK, where she's basically studying evolutionary dynamics in these microbial communities, and where she can actually tell us a lot about what she learned in microbial communities and microbiology and how we can use this in cancer. So Diana, please.
Thank you. Share my screen. Can you all see?
It's perfect.
Okay, perfect. Okay, well, thank you so much for the introduction. As as it was said, I don't work in cancer directly, so my system of choice are microbial systems. We can analyze in the lab quite easily. But since we really use these systems to create physical models that tell us something about how evolution basically occurs in spatial settings, there's a lot of our insight that comes from microbial systems that can be transferred to other cellular populations, like, for instance, cancer. So what my group specifically is interested in is characterizing and quantifying the spontaneous genetic diversity in populations and how this raw material is then used by evolution to create adaptation. So this question, for instance, like the presence of resistant mutations in microbial population is actually quite old. It goes back to Lohr and Delbruck in 1943, who were interested in understanding the occurrence of resistance in bacterial populations. So from a quantitative standpoint, this is equivalent to answer the question really of how likely is it to find a certain number of mutant cells at the end, let's say, of an incubation period. So this is very helpful in applications, both to, for instance, be able to predict whether a drug treatment may be successful or not. And this has obviously implications both in infections, so in bacterial infections, but also, for instance, in tumor treatment. And also, this is the case of somatic evolution, it gives us an idea of what's the likelihood of developing predisposing mutations that can make certain cells more prone to then turn into cancer. So the experiments that Lohr and Delbruck did is really briefly sketched there here on the left. The key point basically of the experiment that they did, the key result was that they were able to quantify this called psi frequency spectrum. So the probability that a mutation that occurs in the population is carried by a frequency X in the population. So what I'm plotting here on the right is in log-log space is this probability that a mutation occurs and the probability that this mutation is carried by at least a frequency X in the population. And what Lohr and Delbruck were able to find is that this distribution is actually a power law. It goes like 1/X, so it has a very fat tail. So there are events that can occur actually quite likely, so-called jackpot events, where mutations can be carried by a very high frequency in the population. The mechanism behind which can be understood by looking at the growth process that the population undergoes during the incubation period. So in their populations, these were well-mixed test tubes, population of bacteria going from around 100 cells to 1 billion cells, where the cells were growing pretty much exponentially. So since in each of these replication replication events characterized here by just a link, there's a certain probability that a mutation may occur. Just so because of the shape of the tree, most mutations will occur towards the end of the incubation period and will be carried by only a few proportion of the population. So that's why the probability is very high at low frequency. However, there's a certain chance that a mutation can occur quite early on in the incubation period and be carried by a large proportion of the population. And this was the explanation of the presence of these jackpot events. So as I said at the beginning, this is the work by Lurie and Delbruck and their distribution is really good to understand well-mixed populations. However, in nature, many populations are actually organized in space. So in the microbial world, we have colonies and biofilms. But as I said, this expands beyond the microbial communities. So in the case of mammalian cells, we have solid tumors. And then even beyond that, we have developing multicellular organisms. And in all these examples on the right, the probability that a cell will replicate or not really depends on where it is and what its neighbors are really doing. So what I'm going to show you next is the work that we did to characterize the genetic diversity in the simplest possible case of these, which are these microbial colonies. So microbial colonies are such that beyond the first initial stage, only the cells that are sufficiently close to the edge are able to replicate because they have sufficient access to nutrients. So what we did, this was at the time when I was a postdoc at Berkeley, as a control, we started several well-mixed cultures from single cells. Each of these cultures was harvested, the DNA was extracted, sequencing the whole genome, and aligned to the reference genome in order to scan for mutations and quantify the proportion of the population that was carrying those mutations. So if we do this over and over for all the mutations, we can get the cell frequency spectrum, so the number of mutations that are carried by at least a frequency X as a function of the frequency. And what we see is that, at least for our control, all our replicates are represented here by different lines, fall pretty well over the Lurie and Delbruck distribution. Interestingly, when we repeated the experiments for colonies, so the only difference between the top and the bottom here is that here's the population, the cells are growing as a colony on an agar plate. We did exactly the same, harvested the populations, sequenced, and then counted the mutations and the proportion of the population that carry that mutation. We got something quite different, that a cell frequency spectrum that looked like this. In particular, what I would like to highlight is that at high frequency, let's say for instance here at 1%, we have almost 10 times more mutations. So the effect of this growth on agar plates is that we have an excess of what the Lurie and Delbruck at the time called mutational jackpots. So the question is, where are they coming from? So we were very lucky at the time that in the lab really next to ours, which was a yeast lab, so another kind of microbial system, a switcher yeast strain was engineered that effectively is able to switch from one fluorescent color to the other. So I would like to highlight this is not a mutation. This is actually a stochastic switch. But interestingly, it can happen only once in the cell cycle. As I said, it's stochastic and it's also inheritable. So that means that from I guess a point of view of a physicist, for all intents and purposes, this is effectively like a mutation with the advantage that we can track this under the microscope. So what I'm going to show you here is a movie starting from a single cell of this yeast strain that is growing over time as a colony to around 10 to the 6 cells. And what you'll see is that it starts purple, and then over time we see this yellow speckles here that represent these switching events that occur during the growth process. If we use the area of these yellow spots as a proxy for the clone frequency, we get again a cell frequency spectrum that has the shoulder-like distribution similar to what we saw in the sequencing data from E. coli. In particular, what we see in this figure is that clones come really in two spatial flavors. There are what we call sectors, which are these clones that have emerged at some point during the growth process and keep growing with the colony. So keep being in touch with the surface growing edge here. Instead, we have other clones like this one here that we call bubble, where at some point the clone lost contact with the front and stopped growing, so it's stuck in the bulk of the colony and is not effectively growing. At the moment it's alive, but they're not growing. So what was interesting in these experiments that we did is that even if they were done on two very different microbial species, one is a bacteria, the other one is a eukaryote, they were still displaying the same kind of side frequency spectrum. Now, the key ingredient that is common to the two is, as I said, this surface growth, so this ability to grow only at the edge where there is available space. So to see whether we were able to capture this high frequency spectrum just by using this simple ingredient, we used a very simple lattice model called the model. So in this model, each cell, each lattice site here represents either an empty site if it's white, a wild type cell if it's purple, and a yellow cell if it's a mutant. So at each time step in the simulation, we pick a random site that has at least one empty neighbor, let's say this one, a random direction in which this site can expand into, let's say the one here on the right, and then with a certain sort of mutation rate, we can switch this site in the new invaded site from purple to yellow. We cannot have the backwards mutation since our history didn't have the backwards switch. So if we do this over and over and over, we get actually a movie that looks more or less like this. So what you see is that this is at least qualitatively very similar to the yeast colony that we saw before. And in particular, in both cases, we get the bubbles that I said and this triangular shaped structures that are the sectors. And I would like to point out here that the mutations here are purely neutral. So the only reason why some clones become bubbles and others become sectors is purely due to stochasticity and to the fact that there's only so much space at the edge that can be filled up. All right, so we can, since we have simulations, we can now do this over and over and over, and we can get a very smooth distribution for the high frequency spectrum. And what we find is that we get really these two regimes as a low frequency, shallower distribution that corresponds to the bubbles, and the high frequency part that instead corresponds to the sectors. We can go beyond this and actually rescale all our sequencing data from E. coli, and we see that they match really well this gray distribution here from the simulation. So it seems that just this key basic ingredient of surface growth is able to capture what we see. As I said, we can go actually even beyond this, and we can model basically the expansion of these clones, looking at the boundaries between the resistant mutants and the wild type. So it turns out that at least in E. coli, these boundaries follow a super diffusive random walk. Then from the statistical properties of this random walks, we can determine a typical width of the bubble and the typical length of the bubble. And if one carries on with the calculation, one can predict actually that this shallower part of the distribution here corresponding to the bubbles is actually a power law with an exponent of -2/5. Something similar can be done for the sectors, and we get here instead another power law with exponent -4. So the shoulder-like distribution that we see here is really the combination of two power laws with different exponents, and the crossover in between the two, which is, whose position is determined by the size, actually, of the colony. Interestingly, so this is in 2D, but if you go in 3D, you get something qualitatively very similar, just with slightly different exponents here. All right, so what you see already from this distribution, so this is in log scale. So what you can see is that most of the mutations, if they occur, they are actually bubbles. I would say 99.9% of them. So the bubbles, as I said, are actually trapped inside. So they are alive, but they're not actually growing. So we can extend our model to actually include some sort of fitness advantage or disadvantage for these resistant mutants. So this was the colony I showed in the case of neutral mutations. But if we assume that, for instance, these mutations are deleterious, as it may be the case for resistant mutations, so mutations that confer resistance, but in the absence of antibiotic, they normally carry a cost, then the mutations will actually be more and more in the bubble category. So if the cost of resistance was high enough, actually practically all mutations would be in the bubble form and we would have no sector. So they will all be trapped inside, here represented by these kind of small yellow dots. So if we now imagine of taking this population that grew in the absence of antibiotics, and then all these resistant clones here represented in yellow, then we take this and we put this colony under different concentration of antibiotic, then we get the following. So, well, if there is no antibiotic, the colony will just keep growing. If there is a lot of antibiotic that kills the purple susceptible cells, then the yellow resistant cells will be able to escape and take over. Somewhere in the middle, there's this like optimal magical concentration that kills some of the wild type, but not enough to completely eradicating it. Then because of competition for space between the wild type and the yellow cells, the wild type actually keeps the resistant cells under control, resulting in a minimal net growth at the end. This was verified in experiments where we inoculated a resistant droplet of resistant cells here in yellow in a bigger droplet of susceptible cells in purple, and let it grow under different antibiotic concentrations. And we saw also in experiments exactly this, this non-monotonic response to antibiotic. So what was interesting, as I was saying at the time, we were coming just from the microbial world. So just reading the literature, we found a paper that came out from the cancer community that came out more or less at the same time that was looking at adaptive therapy in breast cancer, where effectively they described at least as output a phenomenon that was very similar, where the tumor responded relatively well to a low dose of chemotherapy extended over long periods of time, while instead a high dose was initially apparently removing the tumor, but then the tumor would come back and actually be resistant. So this really inspired us, because we thought, okay, well then maybe when we have like solid tumors, then it's something, this kind of spatial distribution of clones might be an example to that would result into this different response to chemotherapy, where actually low doses might be more optimal rather than a high dose of chemotherapy. Okay, so just to quickly wrap up here, so the take-home message from my talk is, I hope I showed you, that just simple spatial growth leads to an excess of high-frequency clones, and this is because only the surface is actually be able to replicate. And again, just simply because of surface growth, these clones come up here with two different spatial distributions, either bubbles or sectors, and the bubbles are really like stuck inside. So if there is an environmental change, for instance, the application of some kind of drug that kills the wild type cells, then the resistant cells may be released, leading to actually an optimal therapy that is somewhere in between, and not actually the highest and most intense therapy that one can think of. So just to quickly mention what we are working on now, again, using microbial systems to get inspired on actually what's happening in this more complex spatial settings. So as I mentioned really at the beginning, in spatial settings, whether a cell grows or not only depends on location, but it can also depend on what the neighboring cells are doing. Maybe some of the most complex environment for microbial systems are actually biofilms. So this is something we're investigating right now. So what I'm just showing you very briefly are two videos of two biofilms, one grown on a liquid liquid air interface, where you see hopefully you can see the aggregation of the bacteria, then the creation of a mat at the interface, and then the formation of these wrinkles that really try to optimize the surface in the biofilm. And this is somewhat different to what instead we see on a solid air interface, where initially we see a growth that is somewhat similar to a colony. But then over time, we see the creation of these wrinkles here that really create channels through which nutrients can go from the outside towards the bulk of the colony, somewhat similar to a vascular system that we would see in a vascular tumor. So we're currently investigating what these kind of complex environment, how this complex environment actually change the genetic diversity of this population. And with this, I'll just end. As I said, I wanted to ask to thank Oskar Halacek and his group, where I did pretty much all the work on colonies, and the people currently in my group that are working on the biofilms together with collaborators. And I'll take any questions that might be. Thank you.
Thanks, Diana. This was a beautiful talk. Also, you've been very, very efficient, which basically means we have a few minutes for questions. And luckily, at least we have a couple of takers. I'll start with Alexander Anderson. So Sandy asks, well, he says, beautiful talk. Did you try adding either death or migration to your model? And if so, how did it impact the results?
Yeah, it's a good question. So for the death, we haven't actually really looked at it in this context. We've looked at it, in follow-up work, we've looked more at the results of mechanical interactions, basically, between the cells. In that case, we have looked a bit at the effect of death. So if death is distributed more or less uniformly, then the results don't change, I guess, qualitatively very much. Obviously, it's very different if death is not uniform, and if some patches are going to be more, for instance, the inside may be more subject to death. So that would be actually interesting to test and see what happens. Regarding migration, this was done not by me, but by other people in Oskar Halacek's group. So what you find in that case, obviously, if the migration is very, very, very high, then you eventually go back in the well-mixed limit. So what is interesting is that there are some well-defined different regimes depending on the kernel that you use for migration, where you get a different kind of side frequency spectrum depending on this kernel, and there are almost like transitions, like clear transitions between these regimes. Yeah.
Cool. Is it fair to say that right now you consider space to be homogeneous? And if so, how do you see things as a change if you actually had like a more heterogeneous environment?
Yeah, that's a very good question. So yes, right now, how we have it at the moment is that space is homogeneous, though I guess nutrient concentration is not homogeneous, which is why some cells grow and others don't grow. Something that we are looking at, not actually with this, not with these microbial systems, but with actually viral systems, is what happens instead if the space, in that case, the space of the host, obviously, is fragmented around and undergoes some kind of interesting dynamics. So it's definitely something on our radar that I think would be very, very interesting to look at. And even in the case of, I guess, microbial systems like biofilms, it would be interesting to like create a more complex, like porous, for instance, environment and see what happens in that case.
Well, I really like when talks are a bit shorter, because that really allows people to ask questions and take the conversations in unexpected ways. So Fred Adler says, beautiful combination of theory and experiment. Could you say a word about why the length of the boundary of the clone follows a super diffusive random walk? Does it follow some kind of death process?
Yeah, that's a good question. So the reason, so it's actually a very, I would say, difficult question in principle, in part to answer, because it's unclear what exactly makes certain bacteria grow, like develop this super diffusive versus diffusive random walks. It seems apparently that just depending on the conditions, for instance, there are a lot of people that work on this, you can get different behaviors. And it's unclear, it doesn't seem to be shape, it doesn't seem to be growth rate. It's a bit unclear actually what happens. It's the intuition is that basically as soon as we get, let's say, just stochastically a curvature, let's say in the front, so there is a part of the colony that is going a bit further, it will then have access, even more access to nutrients, so it will tend to grow even more. So the roughness basically of the growth of the colony edge increases, and then the boundaries really follow perpendicular to this colony edge, which is why there is basically this super diffusivity occurring. However, as I said, it depends a bit on the conditions, so it may become actually diffusive. If it's diffusive, I mean, one can replace basically everything using diffusive exponents, and you still get qualitatively the same behavior, just that the shoulder that I showed before is a bit flatter.
Very cool. Joel Brown, which we all know because I think we are not going to be surprised by this question. Diana, super talker material, your system seems to be quite game theoretic in terms of neighborhood effects between cells and their phenotype. Would game theory be a useful way to conceptualize your system?
I think so, and we haven't done anything in that direction, but I think it would be very, very interesting to try to add some basically game theory into these spatial models and how neighboring cells interact. And I think actually using microbial systems would be relatively easy to play around with this in the lab, since we can very easily genetically modify them to have all kinds of different behaviors.
Cool. Yannick, very short question. In your computer simulations, do you consider growth in 3D?
Yeah, so we did repeat the simulations, the hidden model simulations in 3D to see how the psi frequency spectrum would change. And you get qualitatively something very similar, where you have bubbles and now are three-dimensional and sectors that are kind of three-dimensional cones, basically. So you again get this shoulder-like distribution, and you can estimate the exponents again using the super diffusive random walks, but now you have super diffusive surfaces, I guess, that annihilate. So the result is very similar, it's just again a bit shallower. So the actually intuition is that the well-mixed case is actually the infinite dimensional limit that gives you the complete flat distribution. Mm-hmm.
And I think we have, no, actually, questions keep coming and we have more time, but not a lot. So let's see how many can we go through before Jill's talk. So Lawrence Sasche asks, well, thank you for your great talk. When transferring the bacterial experiments to tumors, one will have to ensure that surface growth actually plays a role in tumors. Mm-hmm. Are there any indications of this?
Yeah, we think, I mean, as I said, I'm not an expert on tumors, but we think that avascular solid tumors where they basically grow more on the surface rather than the core, because the core is not, well, doesn't receive nutrients. It might be a good system basically for this kind of models. Mm-hmm.
I appreciate the conciseness of your answers as well. So Yannick again asks, do you think that this kind of spatial considerations suggest that continuous low dose is preferable to intermittent high dose?
Tough question. Yeah, it's a good question. I don't know. We haven't tried. We haven't tried to see which one. I think I have to say this just very briefly. So there is a caveat in the simulations that I show, is that we basically turn off mutation rate once we apply the drug, which is obviously not true in the actual system. So one would need to consider that actually mutations can keep occurring. Then as they keep occurring, at that point they will be very beneficial because there is the antibiotic, for instance. So I don't know. I'm actually not sure what the answer to this question is.
Very good. And one last one, Sandy, and then we can move on, is in the wild, bacteria rarely grow in these homogeneous conditions. The environment is much more heterogeneous, especially in terms of nutrients as well as space. Yeah. How do you consider nutrient starvation?
Yeah, so in the colony case, as I said, that the starvation, even if the agar is flat, actually you create this, just as the growth proceeds, there is a nutrient gradient that occurs between the edge of the colony and the core. So the core is actually starved. And that's why it's not growing, basically, in our simulations, and everything is going on the surface. But it's true that the way more complex scenarios can occur. So, we mentioned, for instance, porous media would be a very interesting place to go forward.
Awesome.
Okay.
Thank you very much, Anna.
Thank you.
Thank you.